So I'm going to start off with, they requested the learning objectives, and this is mostly so that I make sure that we cover them and we're all kind of on the same page. And this was what was on the flyer for the grand rounds. But in order to introduce these learning objectives and come away with the main themes, I'm going to talk about the variation in von Willebrand factor, and then the key things that I've been interested in heritable von Willebrand factor, both sort of at a high level, and then what we've learned from some of the studies we've been doing, and then come back to these learning objectives. So the most important thing to understand is what von Willebrand factor is. And von Willebrand factor is both complicated and simple in that it is a critical coagulation glycoprotein. It's an adhesive protein. I think of it as like the mucus of the vascular system. It's kind of like a participant in innate immunity in that it's sealing the leaks and recruits inflammatory cells, but is also a critical part of the clotting cascade, and that's how we know it best. The main functions of von Willebrand factor are to stabilize factor VIII, which is the factor that's deficient in hemophilia A. VWF also adheres at sites of vascular injury where it not only probably delivers factor VIII, but also recruits platelets and begins the initiation of the platelet thrombus. So von Willebrand disease is the reason we know about von Willebrand factor, and it's named after Dr. von Willebrand. Because it's the most common inherited bleeding disorder, and the complexities of that we're going to get into later, but that's what brings it to the forefront. And von Willebrand disease is due to quantitative or qualitative deficiencies in the VWF molecule. The flip side of von Willebrand factor is that it also participates in thrombotic diseases, and we know this best for TTP because VWF is a critical component of the pathogenesis of TTP. But high von Willebrand factor levels are also associated with MI and stroke and DVT. So it's really two sides of the clotting coin, and it's important in both. So where does von Willebrand factor come from? It's only synthesized in two cell sites. It's synthesized in megakaryocytes, and it's synthesized in vascular endothelial cells. And we know the most about vascular endothelial cells because we study HUVECs quite a bit. And also, we know from old pig transplant studies that most of plasma von Willebrand factor comes from endothelial cells, so I'm going to focus on endothelial cell processing. So we know that there are genes for von Willebrand factor in the nucleus, and they're going to be transcribed and translated, and this encodes a monomer, a von Willebrand factor monomer, that then is going to go through the normal cell processing pathways. And this monomer is in itself a slightly complex structure. It's formed of multiple repetitive domains, each of which have distinct functions. And we know a lot about the function from the mutations in disease. So D1, D2, this is the propeptide, and this is an isomerase that participates in multimerizing the von Willebrand factor as it's processed. This is the region that binds factor VIII and D', D3. You also have cysteines here that participate in multimerization. And then this whole region here, A1, A2, and part of D3, is encoded by one huge exon called exon 28, which makes it easy for sequencing. And this is also a key area because this contains the A1 domain, which binds to glycoprotein 1b on platelets. And this also contains the A2 domain, which is contains the cleavage site for ADAMTS13. And then there are multiple other domains that participate in other functions, including adhesion to collagen, other secondary actions with platelets, and dimerization. So this is the structure of a monomer, which is missing the propeptide early on as it's cleaved, and I'll show you in a minute. Oh, and I forgot to say, lately it's been proposed to make the structure even more confusing, but I would say that we can stick to the original domains. This new proposed architecture is really just breaking down each of these repeats into mini repeats. And so a D domain consists of each of these elements, and you can see that they're repeated. So I think I understand why, and it's totally true that this is the structure of von Willebrand factor, and I'm kind of hoping for simplicity's sake that we stick with the older nomenclature, and I'm going to use that nomenclature in this talk. So in the ER, the monomer is bound, the two monomers bind together through this disulfide bond in the C-terminal end to form a dimer. And then in the Golgi, the VWF molecule undergoes extensive post-translational modifications, which include heavy glycosylation and also multimerization. So this single monomer is now basically a gigantic polymer, and this processing is very similar to the way mucin is processed as well, which it has great homology to. And this processing continues on as it's targeted to very specialized secretory bodies called Weibel-Palade bodies. These bodies are defined by the presence of von Willebrand factor. You can't be a Weibel-Palade body without von Willebrand factor. And in here, these huge ultra-large multimers are packaged into little coils with the help of the propeptide. And in this organization, you have a very large set of molecules that are basically in rods in Weibel-Palade bodies. And then when you stimulate the endothelial cell, you secrete this ultra-large von Willebrand factor into the luminal side. That said, there is a basal-like secretion pathway for von Willebrand factor that's sort of poorly understood, and it probably is in part incompletely processed and incompletely targeted von Willebrand factor. And that looks like it goes both directions and is probably the source of vascular subendothelial von Willebrand factor. Once in circulation, VWF is protecting factor VIII, although most von Willebrand factor molecules aren't carrying VIII. All of the VIII, conversely, is on von Willebrand factor, and the propeptide goes its own way. We don't understand very well what happens to von Willebrand factor after this, and some of it is probably targeted to carbohydrate-specific receptors, and we know that in plasma, probably under shear conditions, VWF is proteolyzed by the VWF-specific protease ADAMTS13, which can break down ultra-large multimers into normal-sized multimers. So this said, as you can see, it's a complicated process, and lots of differences at each stage of that process can result in variability in von Willebrand factor, and we see huge variation in von Willebrand factor in populations. And you can get this from the normal lab range, which goes from 50 to 150 to 200%, which is a huge range for something that should be so critical. We know that at the low end, you're at increased risk for bleeding, and you're more likely to be diagnosed with von Willebrand disease. And if you're at the high end, and I've put it conservative, greater than 200, but probably greater than 150, you're at increased risk for thrombosis. And the strongest evidence is in MI and stroke. The causes for this variation are multifactorial. And from old twin studies, we know that 1/3 of the variation is due to acquired or environmental factors, and 2/3 is due to inherited factors. And these environmental factors create noise in our research data sets, but are important to remember because you see them in the clinic, and this is, with advancing age, von Willebrand factor goes up. If they are stressed physically, if they're stressed emotionally, if they have syncope from phlebotomy, if they exercised within the last 24 hours, their von Willebrand factor goes up. The hormonal status is important, and some exogenous estrogen formulations can increase von Willebrand factor, and some don't seem to. VWF definitely goes up with pregnancy, and it can cycle with a normal menstrual cycle. So environmental factors aside, there's strong evidence for a heritable contribution to von Willebrand factor level, and of this, approximately 1/3 of the genetic variation can be attributed to ABO blood group. ABO blood group is a significant genetic modifier that comes out of nearly every association study with von Willebrand factor in the genome. And the only exception I'm aware of is in a large Amish pedigree where everyone's probably so closely related, we're not going to find ABO. Multiple other modifiers have been identified more recently in detailed GWAS in large populations, including FUT1 and FUT2, which are precursor enzymes for ABO, as well as STXBP5 and STX2, which are probably involved in vesicular trafficking, and CLECL4M, which is a lectin clearance receptor, and probably there's new data in mice demonstrating that this can clear von Willebrand factor. And then lastly, and I think not at all least, is the von Willebrand factor gene itself. There is tremendous variation in the von Willebrand factor gene, and we know the most of this from studying von Willebrand disease patients. So just to bring it together, von Willebrand factor, extremely complex. We have environmental factors, and in the case of von Willebrand disease, we often have a primary disease-causing mutation, but this is probably modified by all those other things that modify von Willebrand factor in normal, healthy people. So this heterogeneity makes von Willebrand disease diagnostically challenging. If they are have almost no von Willebrand factor and they're bleeding in the classic pattern, that is not the hard patient. It's the hard patients with a minor bleeding pattern or even a moderate bleeding pattern, and their von Willebrand factor levels are in a range that's difficult to interpret. Type 1 von Willebrand disease is the mild to moderate quantitative deficiency in von Willebrand factor. This is the most common form. About 70% of patients with von Willebrand disease will be classified as type 1, and these are where the diagnostic challenges usually lie. Type 2 von Willebrand disease is when there's a defect in the VWF function. So in this case, the common theme is they're having trouble interacting with their ligands. So for 2B, they are too happy to see GP1B on platelets, and so they have atypical platelet binding. 2N has decreased affinity for factor VIII, so their factor VIIIs will be low, and they're distinguished from hemophilia because this is autosomal and not X-linked. 2M, which is very rare, has trouble interacting with GP1B at all. And then 2A is a combination of multiple types that include problems secreting von Willebrand factor and also increased susceptibility to proteolysis. And a common theme in type 2A is that they are missing intermediate and high molecular weight multimers. But type 2, these are qualitative defects where the von Willebrand factor antigen level could be normal, and you find some other defect related to VWF. And then type 3 VWD, which is very rare, and usually these are the results of compound heterozygosity or homozygosity for von Willebrand factor mutations. They really have very little von Willebrand factor, and these patients are not subtle and can present in a way almost like factor VIII deficiency because their factor VIII is also low. So type 1 is the one that becomes quite the challenge. And the type 1 mutations are scattered throughout the gene, as are type 3. Compared to type 2 von Willebrand disease mutations, where the mutations are largely in the domains where we now know they make sense. So type 2N, which changes factor VIII binding, is in the factor VIII binding domain. Type 2B, which it changes the interaction with GP1B, is in the loop that binds GP1B. There's a little circularity in this logic because some of these functions were figured out because of these mutations, but it's later gone on to show that that's what these domains are doing. So the challenge in type 1 von Willebrand disease is, are you looking at a patient with a disease or are you looking at a patient with a low von Willebrand factor phenotype? And you can see that the challenge is related to genetics in that the von Willebrand factor gene mutation patients tend to have very low von Willebrand factor, and then it trails off as your VWF approaches what is the normal cutoff for the lab. But the bleeding phenotype, you have a fair number of people here, let me show you, in this region who you don't find a VWF gene mutation, but they will have bleeding. And it doesn't necessarily mean they have von Willebrand disease, and it doesn't mean they don't. And so a lot of clinical criteria are required for von Willebrand disease diagnosis. And the controversy for a while had been, do you need to have a gene mutation to have von Willebrand disease? And the answer is you don't. So I'm going to take what I've learned from this project working with a fabulous group in the exome sequencing project to illustrate in normals the challenge of figuring out what's going on with von Willebrand factor related to gene variation. And I don't know if any of you know about the NHLBI exome sequencing project, but University of Washington's a major site, and you can find it on the web. I think I have it. Oh, I'll have it on another slide. And the main goal is to take these large cohorts, which have been well phenotyped, and many of them have had SNP data and GWAS data, and to use exome sequencing to see if you could figure out new gene associations and novel disease and phenotype mechanisms using DNA sequencing as opposed to the conventional SNP approach. And I have underlined and to share these data sets because philosophically I feel like this is a critical thing for all research and clinical efforts, is that sharing of data, particularly large data sets, is absolutely essential to understand highly variable phenotypes. So in this, we looked at von Willebrand factor, and we looked at the ethnic differences between European Americans and African Americans and focused on African American gene variants and what their influence was on VWF. And the reason for this was we just don't know a lot about VWF in African Americans, but we had data that VWF in African Americans was different than in European Americans. VWF antigen and factor VIII are generally higher in African Americans, and this is not due to differences in ABO blood group. because we know there tends to be more B or whatever, but this does not seem to be the reason that we have high VWF in factor VIII. And the VWF ristocetin cofactor to antigen ratio is lower in African Americans. So the VWF ristocetin cofactor test is an artifact of the lab, but we use it. So ristocetin was an antibiotic that was being developed, and it turns out it makes you thrombocytopenic, so not a great antibiotic. But the reason it makes you thrombocytopenic is it helps the GP1B integrant on platelets hold hands with the A1 domain of von Willebrand factor, and it is very useful in diagnosing A1 conformations in von Willebrand factor that may be more receptive to seeing GP1B or less receptive to GP1B. But that said, everything depends on the interaction of ristocetin cofactor with these, both GP1B and von Willebrand factor, to be kind of stable, and then to say all the differences are in von Willebrand factor, all the differences are in GP1B. But that said, this is what's usually meant when someone reports a von Willebrand factor activity, is a VWF ristocetin cofactor activity. And recent studies of von Willebrand factor gene variants in African Americans were suggestive that there's a fair amount of variation in von Willebrand factor gene in this population, but the association with VWF phenotypes was unknown. And so there's 2 studies that were recent, one of which was from the Wisconsin group. They only had 66 individuals, and so they were able to say probably reasonably that none of the variants they saw were causing a disease, and that none of the people presented with a bleeding defect. And then the Thousands Genomes Project has a lot of genetic data on von Willebrand factor, but they have no phenotypes. So when doing a genetic study, it's great to explain what the variation is doing, but a lot of the time we only know that the variation is there, and the effect of it only can be done with biological correlates. So our hypothesis was pretty simple, which I like. We hypothesized that von Willebrand factor gene variants are different between African Americans and European Americans, which we felt pretty confident would be true given ancestry. And then the African American VWF gene variants are contributing to the variation in von Willebrand factor and or factor VIII that we're seeing in African Americans. So in ESP, I know that you guys have had a lot of talks from the next-gen sequencing crowd, so I haven't gone much into next-gen, but this is exome sequencing, which is using next-generation sequencing technologies to sequence the coding regions of genes. And so it captures almost all the coding regions in the genome, and is very good at identifying single nucleotide variants and pretty good at identifying small indels and becoming better at structural variation, but that is not something that we were looking at. So we looked at single nucleotide variants and small insertions and deletions. There's exome sequence data at the time that we did this analysis in about 6,500 people. About 4,000 of them are European American descent, and 2,200 of them are African American descent. And of these, looking in the von Willebrand factor gene, which if anyone's ever looked, it's 52 exons and huge. There's a lot of variance, and part of that might be just simply because it's huge, and part of it might be because VWF actually is a highly variant gene. So of these, 452 were in European Americans, 409 were in African Americans, and a substantial number of the African Americans SNVs were much more prevalent in African Americans, and the converse was true in European Americans. So knowing that we have VWF variation, the exome captured captured the VWF gene very well. We then look in the population cohorts because we really want functional data to ask, do any of these variants impact von Willebrand factor? And so in 4 cohorts, there were VWF and or factor VIII information in African Americans. And there was actually pretty good reproducible stability in which assays were done between all of the different sites, which is an issue for factor VIII, which I'm sure you guys know. So the atherosclerosis risk in communities, ERIC study, CARDIA, MESA, and WHI. So the one obvious bias is WHI, that is all women. But other than that, they compared pretty well. And the important thing to know is that these patients, nobody in this study that we know of is in here because they have a bleeding disorder. These are supposed to be healthy population cohorts. So we're looking at what's the variation in a normal population. And I'm sure some of them have von Willebrand disease and we didn't know. But for the most part, these should be healthy people. So in this cohort, there were about nearly 3,000 African Americans which had a von Willebrand factor antigen level, and about 4,400 that had a factor 8C activity by one-stage assay. And you can see here's the average, 132 and 155. These are a little bit higher than their European American counterparts, which is consistent with what was previously observed. So the 409 African American variants that were in the exome sequencing project, 147 of them had a frequency of 0.1% or more, which we would need to really say anything meaningful about an association with a phenotype. And of those, 114 could be successfully imputed in the 4 cohorts. And so this is in part important to remember that we could do this imputation in large part because we had such a large exome data set to be able to fill in the missing data. And it's important to have those big data sets when you do imputation because you don't want to compare apples to oranges with demography. So of these, 30 were missense, and so those are the lowest hanging fruit to actually do a change in von Willebrand factor function. So we looked at those 30 missense variants with von Willebrand factor antigen and the factor VIII data. So 8 of those we found to be significantly associated with von Willebrand factor and factor VIII levels in African Americans. And so this is where they're positioned this is the monomer structure I showed you earlier. And I want to point out that exon 28, which is usually what's sequenced for von Willebrand factor, is here. So not here and not down here. So let's go through them a little bit slower. At 789, if you change threonine, then VWF and factor VIII are higher, actually higher. And this was a very common, like the MAF was almost, actually, this almost was the common variant. And then if you change a residue at 817, factor VIII's low, but VWF antigen is normal. And so that's something there. If you had started with a low VWF level and then added on top of that something that lowered factor VIII further, you might be thinking about a 2N mutation, except we know that we're finding it in normal people. These 2 SNPs were linked, so we can't actually say which one of them is doing anything, if not both. Linkage means they're close together and they're inherited together. So, so in A1, we have variants that increase von Willebrand factor and factor VIII, and 2 variants that decrease von Willebrand factor but don't do anything to factor VIII. And I shouldn't say do, I should say associated with. And then we have these variants down in D4, which we kind of weren't expecting. So a change in residue 2185 is associated with decreased von Willebrand factor and factor VIII, which I think we could all say, maybe it's just changing the processing, it's less efficient. But I cannot explain 2287 yet. And because this one, the VWF antigen levels it was associated with were normal, but the factor VIII is low. But this is nowhere near the domains that we know of that interact with factor VIII. So, and it's not in linkage with any of the things up here. So we don't know why this is associated with factor VIII. And so we were able to look at a dose effect for these common variants. So these are the minor allele frequencies, and you can see that this one is really common. Almost half the people have it. And I think that high frequency lets you see small effect sizes. And so you can see that that you can have this 817 variant that doesn't do anything to von Willebrand factor, but does in fact have a sizable effect on factor VIII. And in addition to these common variants, we also had rare variants. And I'm going to point them out because all of these were associated with a fairly large effect size in von Willebrand factor, decreasing the VWF antigen level by 35 to 40. And that said, I think this also probably illustrates that rare variants really need to have a large effect size before you're going to see them. So there's probably other variants in there that are doing 6 or 9% that we're not going to see just because they're not common enough in the population that we're looking at. And I want to bring this up here. We didn't have ristocetin cofactor data on enough people to really do a very meaningful analysis. But a change at 1472 is also known to be common in African Americans and really negatively impacts the ristocetin cofactor binding activity. And the reason to know about this variant is because these people are normal. They're hemostatically normal. And this is a variant that's sitting under a risto binding site. and it makes it so that Risto doesn't work in the lab. But remember, Risto is an artifact of humanity, and their VWF is fine. So in a person of African American descent who has a really weird Risto finding, this could be the reason, and it's very easy to find out if this variant is there. So the conclusion from this study, and I'm hoping that I'm demonstrating for you guys, von Willebrand factor is really variable, and there's a lot going on in the von Willebrand factor gene. And common and rare VWF coding sequence variants can be associated with effects on VWF and factor VIII even in normal people. And so the VWF gene is probably participating in this spectrum that we're seeing of von Willebrand factor levels. And when it contributes to this genetic complexity, you can come up with additive scenarios by which if you add a non-pathogenic, a non-pathogenic, a non-pathogenic, you may get to a level at which you would identify them to have a pathogenic defect. And just as a pitch for using next-gen sequencing, this also demonstrated the success of being able to use an exome data set and a large imputation into another data set to identify functional variants. So VWF, there are clearly VWF genetic variants that are pathologic, and there's a huge database, and I'm going to refer you to this because, which have been shown to clearly ablate factor VIII binding or clearly ablate GP1B or truncate the gene, or their mechanism is very well studied and known. And then there are these VWF genetic variants of which we are now understanding more about in studies like ours and others that can influence VWF biology but are not necessarily pathogenic by themselves. And in combination, they could cause a problem, or maybe not, but they can definitely influence your laboratory findings. And then there are many, remember we saw 409 variants, there are many genetic variants that are probably have little clinical significance, and we're finding them because the gene is so big, it's easy to change it, or the gene is permissive, or the phenotype is permissive, and we don't know. So coming back to VWF levels being variable, and we talked about the von Willebrand factor gene itself, which is, I think, the easiest to understand how a von Willebrand factor gene would impact von Willebrand factor. But the biggest genetic modifier is really ABO blood group. And so this is where I've been very glad to be at the Blood Center, because I really am loving the story of ABO, so we're going to talk about it. So ABO blood group, in blood group O individuals, von Willebrand factor is very low. And under the threshold of 50, you are enriched for blood group O, and this is also true von Willebrand disease diagnosis patients, particularly type 1, are also enriched for blood group O compared to the general population. And when you get in the high levels of von Willebrand factor level, you are enriched for blood group non-O, and particularly at the high ends, AB. So ABO is a glycosyltransferase that puts on a GalNAc, which is the A residue, or a galactose, which is the B residue. We know VWF ABO influences von Willebrand factor level, and we know that the glycans are present on von Willebrand factor. So that makes it nice in that you can think that it is during post-translational modification of von Willebrand factor in the Golgi by ABO that you are maybe impacting von Willebrand factor. But it doesn't change how much VWF is made or secreted. There's pretty good data that it impacts von Willebrand factor survival. So individuals with blood group O, their von Willebrand factor levels are lower. If you do a dd-AVB challenge, you will find their von Willebrand factor does not hang around as long. A surrogate of von Willebrand factor survival is the propeptide antigen ratio, and that is also consistent with shortened survival. And O can in fact impact hemophilia as well, and you can sometimes see a shorter response with factor VIII in a person who is O. In addition to ABO, there is H. H is a fucose residue that is the acceptor substrate of ABO. It's generated by two genes, FUT1 and FUT2, both of which have been implicated in von Willebrand factor levels as well. So it's a whole system of carbohydrate blood group antigens. We know about ABO because of Landsteiner and transfusion and I'm going to make Zimmering cringe, naturally occurring antibodies. But because of bacterial presentation of A and B glycans, we will all be running around with antibodies to A if we don't have A, B if we don't have B, both if we are blood group O. So O is the absence of A or B, A is A, B is B, A is A and B. And this is the reason we know about ABO, and this is where most of the thinking kind of stops. You're either O, or you're A, or you're B, or you're AB, and that's it. And we know that that's not the case. So ABO is really a complex blood group system. And I think this is a great table to illustrate why ABO is variable. And this is just within blood group A. And this is by no means comprehensive. But what we will see is, so this is the subgroup that the subjects were assigned. And so this is how well it's reacting with antibody or lectin. And you can see the pluses are more, just like usual. You can see considerable variability in how much glycan's detected. And then we also have hints in that it reacts with a lectin, but not the antibody, that there may be epitope changes, and we see hints with that as well in just a minute. You also see differences in tissue-specific expression. I think that this is the case and not necessarily density, but you can find in saliva where if you make H in saliva, you can detect A and H in some individuals, but only H in others. And then the hint that the more supporting evidence that the epitopes are really different is that an A2 individual, for example, can be capable of making an anti-A1 antibody. So you will have a person who types as A who makes anti-A, and it's not because they've made an autoantibody. So A is variable. And this is important for von Willebrand factor as well. And so this is a study from 2007, which included A2, A3, AX. And you can see that, let me find a good example here, that when you have an A across from an O, there's a dose, A2, O is similar to O, whereas A1O, which I'm looking for, here it is higher than O. And we have seen this in our studies as well. So the paradoxical thing is that the presence of A on red cells does not necessarily indicate that you will have the VWF level of the average A, and that A subtypes will often behave like O. Let me move on. So we wanted to look at ABO subtypes in a healthy sibling cohort study, and this was my hint that ABO was really interesting and I needed to talk to Megan. So we were using a 3-SNP algorithm to assign ABO, and this is actually more SNPs than are usually used to assign ABO. Usually you use this one SNP here that is in linkage to an indel in exon 6 of the gene that is the common O allele. And this is what's used in the GWAS that definitely identify ABO to be associated with von Willebrand factor levels in multiple studies. So the reference is A1. If you have this SNP, you're assigned to be A2. If you have this, B, and if you have this, O. And these are both in linkage to indels, which is an important feature. And using this in the healthy sibling cohorts, we also saw an effect of ABO blood group on O, but there was considerable variation. It's statistically significant. We think that it's less pronounced because these are young, healthy people, and large population cohorts often include older people that have acquired higher von Willebrand factor levels. But in the midst of this, I sort of questioned if we had assigned ABO correctly, in part because I was nervous about linking a SNP to an indel, especially over a distance. And what we found is when we phenotyped whole blood that was in this repository for the presence of A and B glycans, that this SNP algorithm was moderately good, about 95% of the time. It assigned ABO type that we also thought was the case. But we saw in about 4.5% of people, the ABO genotype grossly mismatched the phenotype. And remember, you need both alleles to be coordinated to give you the phenotype we're going to predict. O is recessive, AB is codominant. In AO, if we misassign one of those to be A or O and the other one dominates, we will miss this. So at a minimum, 4.5% of the time the genotypes are wrong in assigning the phenotype. And some of them are really wrong. Like these guys are homozygous for the O, and they're putting on A, and someone put on B. And you can find, these are homozygous supposed to be A, but there's some other polymorphism out there that's putting on B. And so you can see that there was no clear pattern as to the mistakes. There were mistakes in every category. So this SNP genotyping algorithm, number one, probably speaks to the fact that ABO has a large effect size because you still saw it in the population cohort, but also speaks to the variation in the ABO gene itself. So ABO is highly variable, more than 200 alleles, and I would say that this and other data support that using SNPs alone is difficult to classify alleles in ABO because there are so many and because indels really matter in the phenotype. And clinically, for transfusion, misassigning a O to a non-O or the other way around is problematic. And in addition to knowing all these single nucleotide variants and knowing that there's so many alleles out there, there's also a lot of variation out there that we haven't yet described. Even though ABO is an old gene and ABO has been sequenced quite a lot, looking in the exome variant server, we can see that, and these are missense mutations or variations. So you would expect that you would have seen these before, and there's a fair number of variants in ABO that change an amino acid that have not been described in the literature or not in the DB-RBC database. So this makes me interested because I started off studying a gene that's really variable, and now I found its biggest modifier is also really variable. So I am very happy, but confused. So O blood group is associated with low von Willebrand factor, and it has its increased frequency in von Willebrand disease, and it's probably because in order to get the diagnosis of von Willebrand disease, you had to have a low von Willebrand factor, and the cycle continues because blood group O has a low von Willebrand factor. And blood group O is starting to look like it might be somewhat protective against thrombotic vascular events. Conversely, non-O blood groups are associated with high von Willebrand factor, high factor VIII, higher risk of MI, post-stent occlusion, second MI, infarct size, pick a thing, CVA and DVT. But within this, it's not just O and A and B and AB. The subtypes matter, and I would think that as we learn more about ABO, we're going to have a spectrum of glycans that look a lot like what we do when we look at other variable phenotypes, and that we've just been binning people together that phenotypically are actually quite disparate. The mechanisms by which ABO influences von Willebrand factor is still really poorly understood, other than I think we all agree that O, VWF, it has shortened survival. And after that, whether or not it changes proteolysis, whether or not it changes interaction with platelets, there's data for and against all those scenarios. So coming back to human von Willebrand factor variation. So VWF's really variable. In the environment and acquired factors, we have many factors that can change von Willebrand factor, but a substantial portion of this variation is heritable. And we have spent a lot of time disregarding ABO as sort of the positive control of our GWAS for von Willebrand factor and acknowledging that it changes, that is associated with changed von Willebrand factor levels, but the mechanism we still don't quite understand. It's been controversial whether or not ABO blood group should be part of a von Willebrand disease workup, because if you have a borderline von Willebrand factor level in their blood group O, or if you have a borderline von Willebrand factor in your blood group AB, do you perceive those patients differently? And some experts say yes, we should, and some experts say no. Right now, the practice is not to consider it in a von Willebrand factor diagnosis, but I'm still unsure as to whether or not that's the correct thing to do. Von Willebrand factor gene variants themselves are complicated. It's a huge gene and lots of variants. And so this variation, in combination with any of these other known variants, and more are to come, that many studies are underway, will come together to make this spectrum of von Willebrand factor variation, which is not just what we see here, von Willebrand factor antigen levels, but variation in interaction with any of its ligands. We know now that there are variants that change interaction with collagen. We know there are variants that change interaction with platelets. There are variations that change its ability to carry factor VIII. It now looks like it interacts with heparins. It may interact with any number of other, osteoprotegrin. There's many other things that in plasma and inside cells and inside granules that it's packaged with that VWF potentially can affect. So coming back to our learning objectives, the key things that I think I want everyone to remember is it's really variable. So environmental acquired is 1/3, but genetic is 2/3. And it's easy to obscure the genetic changes by discounting the acquired. Like, if in a workup you're unsure, it seems reasonable to check again, because some of these things are transient. And to remember, ABO matters and the VWF gene matters. To describe the impact of ABO blood types on von Willebrand factor, and I've shown you that blood group O is associated with lower von Willebrand factor levels than non-O, and the converse can be true as well. But the problem is we don't yet know what to do with that information. And then I felt like I had to add a third one, so name a factor affected by VWF genetic variation. And I want you all to understand that what happens to von Willebrand factor happens to factor VIII. We don't know if factor VIII goes down in clearance with von Willebrand factor, but when you look at GWAS for factor VIII, it's almost all von Willebrand factor. It's almost all von Willebrand factor. So if VWF is not capable of stabilizing factor VIII, then you have a bleeding defect because of the low 8 as well. So just as research acknowledgments for some of the research data I presented.
So Jill, thank you for coming. Just to clarify, so the patients who you had these whole exome sequences of, were they also participants in one of these normal cohort studies?
They were all from the normal cohort studies. Oh, sorry. So were the subjects who participated in the ESP data, were they all subjects of the cohorts in May? Were all subjects from the cohorts. So these were Eric, Mesa, Cardia, and WHI subjects. And all of the samples were done from fairly early draws. But the ages, the WHI subjects were all, I believe, younger than the rest. So I think that was an issue.
So following up on that question, since those are terrific cardiovascular disease follow-up associated with all of these populations, any of your von Willebrand factor mutations or polymorphisms associated with outcomes in those populations, particularly in African Americans?
Yeah, that's a great question, and that's one of the things I forget to repeat the question. The question was, since these are long cohorts which are designed to capture cardiovascular events and other vascular disease, do we see any association with the von Willebrand factor variants, especially since we were looking at African Americans, with any of the outcomes in the study? And the answer is we would like to know, but we don't yet know. So that is one of the projects that we are trying to implement in ESP. And the parent cohorts, I have to give them a lot of credit. You know, they worked very hard to develop these cohorts, and everybody shared. And so I think it's through that kind of sharing that you can answer the questions about small effect size variance. Megan.
Similar question. What about von Willebrand factor and cancer risk? Because there is that link with some like pancreatic cancer with ABO. Did you guys get to look at that yet or not yet?
That's a really good question. I have not looked at that. So ABO is implicated in the risk for several malignancies. And we have entertained the opposite, which now I'm sort of thinking maybe I should have thought of this, which was what's the role of von Willebrand factor in syndromes like Trousseau's, where VWF may participate in the thrombosis associated with the adenocarcinoma. But I don't know. I would presume that the VWF levels would correlate with what we see in other populations, and that since there's an ABO link, that you might find a link with von Willebrand factor. I would be hard pressed to say that it's more than association, but that's an interesting question. Yeah.
So when we're testing people for von Willebrand factor in the laboratory, should we stratify their reference ranges by their ABO status to be able to say whether someone's likely or unlikely to have a von Willebrand variant?
Yeah.
Contributing to their phenotype? I
would really love to say yes, but the question is, should you be using ABO information to stratify the ranges of von Willebrand factor in the lab? And I would really love to say yes, but it's just like every other von Willebrand factor. There's considerable overlap between any of the ABO groups. And so I personally, I think the AB and the O are far enough apart that you might be able to say that someone with AB and 45 is really getting out of their range. But there's so many other things going on I kind of think it might be one of those things where you punt and you say, here's their ABO blood group and here's their von Willebrand factor, and do with it what you will. But it was considered I think it was considered in subcommittee at ISTH, and I think the answer was no. But I think it's because you have so much variation. And I think part of that may be that we have not been precise enough in assigning ABO.
I mean, sometimes when we're signing out von Willebrand cases, we have these sort of borderline low people. We speculate that maybe they're type O, but we don't know. Though we have a laboratory now at Harborview that can help determine that.
So you'll have variants though that are normal, that are hemostatically normal, but have decreased reactivity with ristocetin, just as a.
Yes, that's well established.
So there will be variants out there who, by our typical diagnostic tests, have polymorphed away from diagnostics to make them look as though they have a disease where they don't, in the context of a disease that's reported to be incredibly frequent. So based upon your genetic state, is there any ways of predicting the frequency of polymorphisms in ZWF where they are decreased ristocetin activity but normal hemostatic function?
Well, for that particular, I think the summary of the question is that that when you have a variant in von Willebrand factor that you know is giving you an abnormal lab test that you think is not significant in vivo, is there a way to use the information we understand from VWF gene sequencing to predict?
What's the frequency of that?
Oh, so we know the frequency of these alleles. So most of them are pretty rare, but you're going to see them. So they're in the 0.1 to 1% range. It's the really rare ones where I think, and this is my sort of bid for sequencing, the rare ones need to be worked up so that we know what to do with them the next time we see them, which is infrequent and but collectively common. So we have the fortune of Veronica and the Wisconsin group having worked up that particular Risto variant and demonstrating that it is really not causing bleeding in the patients. Because you will have a patient who presents with bleeding, you find a low Risto, and you think you're done, and then the family doesn't make sense because you have a person who doesn't bleed. In fact, maybe they clot or something, and their Risto is also extremely low. And so it's those hints that need to be investigated each time. So for that particular variant, we know what to do. If you see that variant, you know you can disregard it, and the Risto isn't your test of choice. But beyond that, each variant really needs to be evaluated separately.
Another question. So I always forget this. VWF carries ABO glycans.
Von Willebrand factor has ABO glycans both on N-linked and O-linked sugars.
So we give out a group plasma routinely. So the half-life of VWF from an A donor into an O recipient is the same or different than the half-life of VWF from an O donor into an O recipient?
There's a little data to suggest it's not as good in that it lasts shorter. And this was the origin of asking for blood type specific cryo before there was other product. That people felt as though they were giving a product that was inferior because it may not live as long. But we certainly give a lot of factor that has the converse glycan on it, and no one's asked the question since then because it lives long enough to provide hemostatic value.
Has anyone asked the question if it fixes complement due to agglutinins interacting with the protein? And anaphylatoxins get released at some low level?
No, not to my knowledge. How would it fix complement?
Well, because if you have anti-AIGM floating around that gets conformationally changed by encountering A on VWF, one IgM is sufficient to get C1q going.
Where would it fix, though?
Well, it would bind to the surface of the protein. The antibody would bind to the protein.
Right, and then where would the complement go?
The complement would be, the cascade would be initiated by the conformational change of a single IgM that had bound its...
Oh, I see. So you're initiating it, and then generally anything else in the environment could get creamed, I guess.
Well, complement does stuff other than lysels,
Right. So no, and I would really love to ask the question, what are the VWF glycans on, what are the A glycans on von Willebrand factor doing? And we have profiled von Willebrand factor glycan burden in our lab, and we see almost no A glycans on A subtype individuals and lots of A glycans on A1. So I would think subtype matters there as well. And that phenotype is also the profile of platelets, which we know that A subtypes are not putting their A glycans efficiently on platelets where A1 is robustly doing it. And I think that a lot of that is substrate preference. A1 is just a more promiscuous enzyme, and A2 is more finicky. So I think the same question could be asked with platelets, and I also would love to do that. And I'm also would like to know if von Willebrand factor is the reason that platelets are presenting A differently.